Morning, everyone. Welcome to SBIA 2021, Advancing Generic Drug Development, Translating Science to Approval. Today, my presentation will focus on use of artificial intelligence to facilitate the development and regulatory assessment of complex generic drugs. This presentation reflects the views of the author and should not be construed to represent FDA's views or policies. Here's the outline. First, I will introduce the definition of AI and its prevalence. Second, I will discuss the opportunities offered by AI for facilitating generic drug development and regulatory assessment. As the third and fourth points, I will talk about our efforts to answer the opportunity call along with actions from other stakeholders. Lastly, I will summarize the learned challenges and the lessons and the final takeaway. AI is everywhere. But first, what is AI? In the 1950s, Alan Turing, the father of theoretical computer science and artificial intelligence, proposed a famous Turing test. Turing proposed that a computer can be said to possess AI if it can mimic human responses under specific conditions. As shown in the right side figure, A referred to a computer and BC represented two humans. During the test, C functioned as a questioner while A and B functioned as a respondent. If after a preset number of questions, C cannot determine the difference between A and B, then the computer A is considered to have AI. Another definition comes from John McCarthy, one of the founders of the discipline of artificial intelligence. He said, "It is the science and engineering of making intelligent machines, especially intelligent computer programs." The recent decade has witnessed the exponential growth of AI. Truly, the thriving AI community should thank the advance in information technologies and powerful chips, including the arrival of a big data era, advanced data analytical tools, for example, machine learning and natural language processing, as well as high-performance computing, HPC, and cloud computing. Indeed, AI is transforming many areas of everyday life, like a smartphone we are using every day, autopilot, chatbot, which has been used for customer service by many companies, personalized recommendation. I believe many of us already receive this type of a recommendation by some manufacturers, ATM, which we are very familiar with. Given the prevalence of AI, we also see that AI offers opportunities to facilitate generic drug development and regulatory assessment. The first aspect that AI can help is to automate labor-intensive works. For example, facilitating bioequivalence assessment. With automated process, we expect to see enhanced efficiency, like saving time, improved consistency, for example, reducing human error, and also high-quality deliverables. The second aspect we see the benefit of using AI is to borrow recently developed advanced data analytical methods. Those technologies can be used for promoting business intelligence, like modeling optimization business process. For that, we will provide a case study in the following content. The advanced quantitative analysis method can also be used for supporting regulatory assessment. For example, assessment of active pharmaceutical ingredients sameness. This slide shows our efforts, the efforts from Division of Quantitative Modeling and Methods under the Office of Research and Standards in OGD to answer the opportunity call. First, it is about automating labor-intensive tasks. We are having two ongoing projects in this regard. One is about the development of a bioequivalence assessment tool, aiming to streamline the labor-intensive but less human intelligence-involved task during the bioequivalence assessment. Another project aims to develop a data analytical tool based on machine learning and natural language processing to facilitate the product-specific guidance development. Moving to the right side, we also conduct research to employ the advanced data analytical methods, for example, machine learning, for promoting business intelligence. Here, I list 3 ongoing projects from this perspective. They are prediction of abbreviated new drug application ANDA submission, heterogeneous treatment effect analysis, and modeling the process of ANDA assessment. Those data analytical methods, for example, multivariate analysis method, can also be used to support regulatory assessment. 2 examples I list here are equivalent assessment of a complex particle size distribution based on the Earth's mover distance, and the multivariate analysis method to facilitate active pharmaceutical ingredient sameness assessment. In the following slides, I will use the prediction of ANDA submission as a case example. In this case example, I will briefly introduce our previously published work, Predictive Analysis of First ANDA Submission for New Chemical Entities, NCE, based on machine learning methodology. Before introducing the study, I want to clarify an important term, NCE. An NCE means a drug that contains no active moiety that has been approved by FDA in any other applications submitted under Section 505B. A 5-year period of exclusivity is granted to NCE drugs, which means that no ANDA may be submitted during the 5-year exclusivity period. An ANDA may be submitted 1 year prior to the NCE exclusivity expiration if they contain a certification of a patent invalidity or non-infringement, paragraph 4 certification. Therefore, ANDAs that reference an ROD with NCE and contain a paragraph 4 certification, the earliest lawful ANDA submission date, in short, ELAST, is 1 year before the NCE exclusivity expires. Here's some background information for this project. An approved ANDA is required to ensure that generic drugs are available to facilitate drug availability and accessibility to U.S. public. Under GDUFA 2, the ANDA assessment process involves multiple offices in FDA, multiple disciplines, and tight turnaround times. Predictive analysis of ANDA submission will critically inform resource allocation and workload management, including but not limited to prioritizing product-specific guidance, PSG development and research efforts, and also optimization resource allocation, for example, pre-ANDA interactions. So in this study, the scope will be time to first submission for ANDA referencing NCE. In order to conduct the predictive analysis, we had to collect relevant data from 3 categories. First category is about drug product information, including indicators for complex API, complex dosage form, complex delivery route, complex drug-device combination. Also, the indicators for abuse-deterrent formulation, oral modified release, anatomical therapeutic chemicals, ATC, and also the indicator for acute and chronic disease. Second category is about regulatory information, including NDA approval date, NCE exclusivity expiration date, patent expiration date, first AN submission date, first PSG publication date, and an indicator for risk evaluation and mitigation strategies, RAMS. The third category is the pharmacoeconomical information. Here we use the drug sales from 2011 to 2017. Among the collected data, many of them are time constant variables, like most of variables for drug product information. However, there are some variables that are time sensitive. This figure shows the definition of several important time sensitive variables. Let's read this figure from left to right as time goes. The first vertical line, we see NCE approval day. As we above described, 4-year post NCE approval day is the earliest lawful ANDA submission day, in short, ELAST. Based on the ELAST day, we extract the product-specific guidance and the drug sales information at 1 year before the ELAST day. And starting from the ELAST day, we predict the time span of first ANDA submission. As shown in the below equation, our analysis can be considered as a modeling time span from first ANDA submission day to ELAST day as a function of drug information, regulatory information, and pharmacoeconomical information. Given the organized data, we conducted the analysis using a machine learning-based survival analysis, random survival forest, in short, RSF. The RSF is a data adaptive method, meaning no assumption for data and model. The RSF is also be able to handle large feature problem, where the number of variables is greater than the number of observations. With this, we can see RSF is suitable for analyzing complex real-world data. The right figure shows how this model is trained and conduct prediction. As shown in the green block, during the training, RSF model is fed both predicted variable and dependent variable as input and output layer. And RSF will iterate the learning process of mapping from input to output and complete the training until the difference between predicted output and the true output reach a preset threshold. As shown in the black block, during the prediction, the predicted variable of test data is thrown into the input of the trained RSF. Then the RSF will provide his prediction. In order to evaluate the predictive performance, we apply Concordance Index, in short, C-index. Briefly, when C-index equal to 1, it means the model has a perfect prediction, while if C-index equal to 0.5, it means the model's prediction is just like a random guess. This slide shows two key results from this study. The left figure shows the comparison between predicted survival curve, shown as a green curve, and the true survival curve, that is a Kaplan-Meier plot from the real data, shown as a black curves. We can see that the predicted and the true survival curve overlapped very well, and also C-index value was calculated as equal to 0.767, which indicate this is a fairly good prediction. The right figure shows the identification of important variables. Machine learning methodology are always criticized to work as a black box and the lack of clarity. The identification of an important variable can improve the model clarity. As shown in this figure, we can see the first three important variables are sales 1 year before ELAST day, availability of a PSG 1 year before ELAST day, and the index of a complex API. Also, the plus minus symbols right after these three variables refer to the positive and negative relationship between this variable and the dependent variable. For example, for the first identified variable, we can see the drug sales is positively related to the earlier ANDA submission. After introducing our efforts, In the following slides, I would like to provide an update on some actions from other stakeholders, which can be reflected by a brief summary of a special session focusing on utilization of AI in a FDA-hosted generic drug science and research initiative public workshop. This slide shows the session design. The session title is Exploring Opportunities and Challenges for Utilizing Artificial Intelligence, for example, machine learning and natural language processing to support generic drug development and application assessment. This session consists of 3 presentations and a 30-minute panel discussion. For presentations, the speakers came from academia, pharmaceutical industry, and the consulting industry. They all presented their pioneer works of using AI to facilitate generic drug development and provided many insightful ideas and comments. The following panel discussion invited presentation speakers and another 5 experts working in this field. You will find more information by checking the link at the bottom. In the next 2 pages, I will provide a brief summary on the presentations and the panel discussion. The first presentation was given by Dr. Yang from the University of Macau. His presentation focused on the challenges and the values provided by AI for drug formulation development. He's also provided a case study which is predicting physical stability, in vitro dissolution, and the in vivo performance of solid dispersion. The second presentation was given by Dr. Oprah from Sandoz Pharmaceuticals. She presented a study of applying AI models to predict BE outcome based on relevant information, such as dissolution, particle size, solubility, and so on. In her presentation, Dr. Oprah also discussed other opportunities of using AI model for generic drug development. Mr. DeLiberty, the president of Montclair Bioequivalence Service, conducted the third presentation. He emphasized the importance of AI to generic drug development, foresaw several potential development directions, and stressed the need to develop regulatory framework for AI. Moving on to the panel discussion, the discussion started with a priority order of the potential application, especially for the low-hanging fruits. All the panelists joined the discussion and provided their insights and comments. For example, handling outliers could be a feasible application by applying AI models. Subsequently, the discussion switched to feasibility from the technical perspective, including combination of AI models and the conventional tools, for example, PBPK model, importance of integrating domain knowledge, and the opportunity of leveraging AI to utilize data from different resources. The discussion also covered other critical aspects from this topic. For example, transparency, interpretability of the AI models, and the business model of collaboration, for example, between industry and the FDA. With above-mentioned works, this slide summarizes the learned challenges and lessons. They are availability of reliable data, incorporation of domain knowledge, enabling complex projection between complex data sets, transparency, interpretability, which is how to turn the black box into gray box, extrapolation ability of AI models, lack of guidance for regulatory purpose, last one, teamwork and collaboration. To wrap up my presentation, here are takeaways. AI technologies bring opportunity to advance development and the regulatory assessment of generic drugs. AI technology also have been applied to facilitate BE assessment, PSG development, business intelligence, and regulatory assessment, etc. Lastly, AI technologies will play more important role in providing high-quality generic drugs for U.S. public, as more challenges get addressed. That concludes my presentation. Thank you for your attention.
Thank you for the nice introduction. Good morning, everyone. Today, I will present model integrated bioequivalence approach for complex generic drugs. The focus of the presentation will be on discussing common deficiencies in pre-ENDA meetings and then talk about good practices. We will use long-acting injectable drugs as an example of complex generic drugs in this presentation. Before we begin, this is the disclaimer that this is the presenter's view and does not represent the FDA's views or policies. Here are the learning objectives today. We will understand the background and challenges in generic long-acting injectable drug product development as an example of complex generic drugs. We should be able to describe model integrated bioequivalence approaches and then describe common deficiencies in pre-ENDA meeting packages involving model integrated BE approaches. Long-acting injectable drug products are intended to release drugs over an extended duration of time, like over days or even months, when injected via intramuscular or subcutaneous routes. They can help improve compliance in patients, and generic product will provide more treatment options and help reduce overall healthcare costs. The challenges in generic long-acting injectable drug development mainly comes from their long apparent half-life. Compared to typical oral drug product with half-life on a scale of hours, long-acting products usually have half-life on a scale of weeks. Typical single-dose crossover study is therefore not possible. In general, multiple-dose crossover steady-state studies in patients are recommended, which is impractical to conduct. If there is no safety concerns, for example, contraceptive long injectables or implant, a single-dose study is possible. But parallel design together with high pharmacokinetic variability, this will require a large sample size. In addition, higher dropout rate is also an issue associated with long study durations. Modeling and simulation can help overcome challenges in generic long-acting injectable drug development, mainly by helping reduce study duration and/or sample size, making bioequivalence studies more feasible. Today, we highlight model integrated method for generic drug product development. This involves using models not just to plan the study, but to generate pivotal evidence for bioequivalence to support product approval. Model integrated approach supports product approval by a pre-specified model-based analysis of an in vivo bioequivalence study. This can increase success probability of bioequivalence by providing a way to quantitatively evaluate the study design. In general, virtual bioequivalence studies are part of model integrated approach, and they are generally pre-specified in the modeling analysis plan, what is also referred to as MAP. Together with in vitro studies, model integrated evidence can provide alternative approach to PK/PD, PD, or comparative clinical endpoint bioequivalence studies. To help us discuss common deficiencies in pre-ENDA meetings involving model-integrated approach, here is an example of model-integrated bioequivalence strategies developed for long-acting injectable product by Uppsala University under GDUPA research contract. This framework directly applies modeling and simulation during the bioequivalence analysis procedure. The general idea here is that population PK model with treatment effects on all absorption parameters because they reflect formulation differences between genetic and reference product is fed to the data from an innovative design study such as a multiple dose crossover switch study, which has a shorter duration than conventional steady-state design. Given the complexity of absorption models and the need to evaluate the treatment effect on those, model averaging was used to address model uncertainty. Then population simulations are conducted using the estimated model parameters. Note that the simulations are conducted using a typical single-dose crossover BE study in this framework. Then, across all population simulations, 90% confidence intervals of the geometric mean of metrics of interest, such as Cmax AUC, are calculated and final bioequivalence conclusion is made. Type I error is controlled in this framework, providing confidence in using model-integrated evidence to support genetic long-acting injectable product approval. Now, we will transition to pre-ENDA meeting experience. Several innovative approaches proposed for genetic long-acting injectable product development by genetic industry. As shown in the example we just discussed, given the complexity of the model-integrated bioequivalence analysis, we encourage you to ask early interaction with agency and exchange ideas with FDA project team. We've seen that information request is common, which takes time away from the assessment clock. To confidently apply model-integrated approach for bioequivalence assessment, the proposed approach should meet the regulatory expectations. It should be capable to detect formulation differences, which is related to type I error, and also needs to show reasonable passing rate for bioequivalent product, which is related to type II errors. From agency's perspective, control of type I error is more important. Model also should be sufficiently validated for the intended regulatory purpose, and uncertainty of the model parameters should be assessed as well as their impact on the bioequivalence determination. The modeling analysis plan, map, and study design should be pre-specified. A common theme we are observing in the pre-ENDA meeting packages involving model-integrated approach is lack of necessary details for assessment. It should be clear what question you want to address using model-integrated method. And once the question is defined, then propose how you will use modeling to convince FDA. Examples of such details include, but not limited to, detailed plan, simulations to demonstrate to FDA how your plan will address the question. Of course, an important detail should be included in the package is how you will justify the proposed modeling approach to provide confidence in using model integrated approach for bioequivalence determination. Some of the deficiencies are listed here. Clear executive summary should be provided, and that can help reduce information request. We recommend you submit the model codes at the time of meeting package submission. If you encounter any issues in submission, prompt communication with us would be recommended. The common deficiencies we discussed can also provide insight into good practices for a pre-end meeting package involving model integrated approach. Here is the first challenge question. Which of the following statements is not considered as good practice? Please select one, and I will wait a few seconds for you to select the answers. A. Clearly identify the question that model integrated approach will be used to answer. Outline the scope of the question and what modeling exercise will be used to address those questions. Provide a succinct executive summary. State the objective model of model integrated approach as general as possible. The answer is D. The objective of model integrated approach should be as specific as possible in your submission. Let's do another challenge question about good practice. Which of the following statement is not considered as good practice? A. Include step-by-step guidance on performing simulation and calculating pharmacokinetic metrics that is submitted in the study report. Submit the code to perform simulations as presented in the study report, including model codes or control streams and output listings from the simulations, as well as the code to calculate the exposure metrics such as AUC, Cmax. Assume there is no need to provide reference to the specific model code simulation output in the study report. Submit the summary of model codes files in the package. The answer is C. Please provide reference to the specific model code simulation output in the study report. Let's change gears and talk about challenges in applying model integrated approach for bioequivalence and how we are making efforts to address those. We discussed regulatory expectations when using model integrated approach for bioequivalence, such as type 1 error control, model validation, uncertainty assessment, modeling analysis plan pre-specification, etc. What additional considerations should be needed than other than the existing guidance on modeling and simulation? We acknowledge that different scenarios may need a different type or level of validation. Here we provided some example scenarios that would require different type of validations. Here are additional scenarios potentially used for model integrated BE proposals that would affect types of validations. Another challenge is related with modeling analysis plan that should be pre-specified. Then which part of the modeling analysis plan should be pre-specified and where post hoc analysis can and or should be allowed can constitute a challenge in the model integrated approach proposal. FDA, in collaboration with Center for Research on Complex Genetics, referred to as CRCG, is planning a public virtual workshop in November this year on best practices for establishing the suitability of a model integrated approach to demonstrate the bioequivalence of long-acting injectable product. We believe it is timely to engage experts in the field of modeling and simulation in the new and generic drug industry, academia, and relevant stakeholders to explore, identify, and recommend best practices for the development and assessment of model integrated approaches for bioequivalent assessment. A particular emphasis will be on how model integrated approaches can be used to support innovative study designs and data analysis for bioequivalence assessment, and how they can be validated and verified. Fostering collaborative development of these best practices could contribute to the availability of more long-acting injectable generic drug product for the American public. To support the workshop, CRCG funded 2 modeling and simulation projects to conduct and present model integrated bioequivalence analysis for long-acting injectables. They will develop and present the best process to implement the modeling approaches using the case examples. Here is the summary of the presentation, and I hope you all achieved the learning objectives we discussed at the beginning of the presentation. Development of long-acting injectable product is often challenging, but model integrated evidence approach can save time and resources in development of long-acting injectable product, making the bioequivalence equivalence studies more feasible for this type of product. Pre-end meeting submission applying best practices can increase assessment efficiency and efficient generic drug development. We shared the workshop that is planned in November information with you. And last but not least, FDA welcomes innovative alternative approaches to demonstrate bioequivalence for complex generic drugs. I would like to thank you for your attention, and I would like to emphasize again that we encourage you to engage with agency only with pre-end meeting request with your modeling proposals. Here are some resources that you can check it out. I would like to thank my colleagues in DQMM, Dr. Satish Sharan, and the division leadership, Drs. Liang Zhao and Lucy Fang, and the quantitative clinical pharmacology team, And for strategic advice from the ORSIO, Drs. Robert Lionberger and Lei Zhang. And also want to thank the external collaborators at Uppsala University, Dr. Matt Carlson and Andrew Hooker and their team. Thank you for your attention.
Good morning, my name is Huzeyfe. Today I will talk about Cryosem for characterization of complex drug products. I am a staff fellow at the Office of Testing and Research in Office of Pharmaceutical Quality. So we'll start with pharmaceutical quality first. A quality product of any kind consistently meets the expectations of the user. Drugs are no different. Patients expect safe and effective medicine with every dose they take. Pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defect. It is what gives patients confidence in their next dose of medicine. This talk will focus on size and morphology analysis of nanomedicines using electron microscopy at cryogenic temperatures. To do that, I will try to give a little bit of a background in nanomaterials. We'll discuss commonly used particle sizing techniques. We'll try to establish a fundamental understanding of what is measured with each technique, as this is important for physical-chemical characterization as well as method development. And we'll finally discuss cryofixation approaches for sample preparation in electron microscopy. Many diagnostic and therapeutic advancements have been made by implementation of nanotechnology in medicine. Over 50 nanomaterial-containing drug products have been approved by the FDA so far. FDA considers application of nanotechnology when a material or an end product is engineered to have at least one external or internal dimension or surface structure in the nanoscale range, that is 1 to 100 nanometers, or when a material or an end product is engineered to exhibit properties or phenomena that are attributable to its dimension, even if these dimensions fall outside the nanoscale range up to a micron. In general, application of nanotechnology has been through conjugation of a nanomaterial with an existing active pharmaceutical ingredient to alter its pharmacokinetics and pharmacodynamics, to increase its potency, decrease its toxicity, and improve the delivery of the drug product. To do that, nanomedicines have to be manufactured with precise size, shape, composition, and surface chemistry. And to ensure that, we have to have analytical methods and techniques that can accurately provide this information. It's important to take a step back and understand why size and shape are so important, what is so special about the nano range, because we know that in bulk, in macro scale, size and shape do not define or do not affect material properties. The short answer is the surface area to volume ratio. Consider a nanoparticle that's 2 nanometers in diameter. 62% of the atoms on this nanoparticle are at the surface. When we come down to 100 nanometers, this ratio goes down to 1.6%, and it becomes negligible when we get into the macro range. So that tells us what is so special about the nanoparticles, because surface atoms behave differently, and bulk material properties are no longer valid in the nano range. One of the best examples that exhibit this effect is metallic nanoparticles. As we know, gold in bulk has a specific color that does not depend on its size or shape. When we come to the nano range, the color of gold nanoparticle solutions depend on the size of the nanoparticle. In a more pharmaceutically relevant example, distribution of surfactant varies significantly in nanoscale in a nanoemulsion, which in turn can affect the distribution of the drug product. For instance, when oil globules are small and as small as 10 nanometers, the surfactant mainly is on the surface of these globules and much less in the bulk, in the aqueous environment. And that affects the distribution of the drug, and the surfactant ratio on the oil becomes negligibly small when we move to larger oil globule sizes. When it comes to shape, we can continue with the same example. Gold nanoparticles that are in rod shape again, the color of the colloidal suspension depends on the aspect ratio of these particles. This is not the case in bulk, but in nanoscale that is the case. In a more pharmaceutically relevant example, for nanocrystalline active pharmaceutical ingredients, cellular uptake and internalization can depend on the aspect ratio, as well as other parameters, but a specific aspect ratio may be preferred for cellular uptake. So, having established that, we can move on to analytical considerations. And when we talk about size and shape, the easiest thing to do would be to look at it with the light microscope. But we have to consider the resolution. Due to the wave nature of light, the resolution, which is the minimum distance to distinguish 2 points on a specimen, is given by this formula. When light passes through an aperture, diffraction occurs, and the result is a bright central region and concentric rings with decreasing intensity. This is called an airy disk. This formula here is for lateral resolution. There are also other criteria such as the Rayleigh criterion and to consider for angular and axial resolution. But what this really tells us is that there's a minimum size that we can see with light microscopy that depends on the wavelength of light, as well as the numerical aperture of the objective. And this numerical aperture in oil immersion objectives can be theoretically as large as 1.5, but realistically 1.4. And that is because this formula has the refractive index here. And for dry objectives, this numerical aperture is less than 1. And that means at 550 nanometers in green light, resolution is larger than 200 nanometers. But realistically, this is a theoretical number. Realistically, we're looking at 1 to 5 microns to distinguish a particle. So that really eliminates light microscopy when it comes to nanomedicines. An alternative is electron microscopy, where the wavelength of electrons is much smaller than that of photons due to the de Broglie relationship. In fact, it depends on the potential that's applied to electrons. It's only 4 picometers at 100 kiloelectron volts, 12 picometers at 10 kiloelectron volts. Of course, there are different types of interactions that occur between electrons and materials. Auger scattering, characteristic X-rays, scattered electrons, secondary electrons, backscattered electrons, and transmitted electrons can be detected. The original form of electron microscopy was in transmission modality. In transmission electron microscopy, the sample, a beam of electrons pass through the sample, and that's how an image is constructed. In scanning electron microscopy, the beam is focused on a specimen, and the specimen is raster scanned, and that's how an image is constructed. There are also other modalities such as the scanning transmission electron microscopy and scanning tunneling microscopy. Here's the schematic of the scanning electron microscope. There's an electron source. There are coils used to focus the electron beam, and the electrons that scatter from the specimen are detected. And it's important to notice that this whole process happens in vacuum. In scanning electron microscopy, in SEM, the resolution can be as small as 0.4 nanometers, but realistically it is 1 to 10 nanometers. But this is a good enough resolution to make size and shape measurements in characterization of nanomedicines. So let's talk about image-based quantitative analysis of size and shape in electron microscopy. This is a scattering electron microscopy image from a protein nanoparticle. This image can be segmented, and that allows measurement of a variety of parameters such as the area, perimeter, and ferrite diameter, which is the maximum distance between two parallel tangents. A circle equivalent diameter can be calculated from area, and these two diameters, ferrite diameter and equivalent circular diameter, can be compared, and that gives us an idea about the deviation from spherical shape in this product. We can also calculate aspect ratio, roundness, and sphericity, and that also tells us about the morphology of particles in this formulation, and that gives us quantitative information about size and morphology altogether. And there's a wealth of information that can be obtained from image-based quantitative analysis. There are also other particle sizing techniques, and it's important to survey them. One of them is called static light scattering, laser diffraction. In laser diffraction, particles are passed through a beam of light, and scattered light is detected at large angles particle size can be estimated using intensity and angle measurements through Fraunhofer-Mie theory. In this case, refractive index of the particle must be known. A equivalent spherical diameter can be calculated, but assumptions in particle configuration and volume must be made, and there are issues with reproducibility and repeatability due to flow and thermal or model-related artifacts. Another technique is called DLS, dynamic light scattering. In DLS, colloidal suspensions where nanoparticles do not settle over time are characterized by Brownian motion. In DLS, this property is used to estimate hydrodynamic radius. Continuously mobile particles cause destructive interference or constructive interference when a beam of light is passed through a colloidal suspension. And this fluctuation in scattered light intensity over time can be correlated against short decay intervals, and the autocorrelation function can be related to the diffusion coefficient, which in turn can provide an estimation for the hydrodynamic diameter with a spherical assumption. But there are issues with sample preparation. Solvent viscosity is very important. Concentration of the colloidal suspension is very important because there can be multiple scattering events, and that must be avoided. Fluorescence can be an issue. Agglomeration of particles can yield incorrect results, and the shape is also critical because there's a spherical assumption. There's an excellent survey on characterization of nanomedicines. Specifically when it comes to particle size measurements, up until 2015, 48% of the applications had dynamic light scattering, 30% of them had static light scattering, 14% of them had microscopy, and 8% of them had another technique such as size exclusion chromatography or asymmetric flow field fractionation. So far we went through light scattering methods. We saw that they provide quick and easy indirect measurement of size, but there are significant limitations with particle shape, sample preparation, and model parameters. It's important to remember, for instance, in DLS, concentration of the sample can be problematic, and dilution of samples in nanomedicines can also be problematic for the sample, as stability issues can arise. In imaging, there is tedious sample preparation, which we will see in the second part of this presentation. Analysis volume is small, but image-based measurements in morphology analysis, it provides a wealth of information. So with that, we'll move on to the first challenge question. Here we're talking about a hypothetical drug product. The question is, which technique is best suited for characterization of a high aspect ratio nanocrystal formulation with particles smaller than 50 nanometers in size? And if we have to be specific, we can assume a particle that has aspect ratio larger than 2 and the D90 is less than 50 nanometers. Options are A, optical microscopy, B, static light scattering, C, dynamic light scattering, and D, transmission electron microscopy. So the answer is transmission electron microscopy. There are two clues here. One is the aspect ratio is larger than 2, which means shape can become an issue with light scattering techniques. And we're looking at particles that are smaller than 50 nanometers, which means optical microscopy cannot be used. So the answer is transmission electron microscopy. So now we're going to switch gears and talk about sample preparation in electron microscopy. The easiest thing that can be done to image in nanomedicine, a colloidal nanoparticle suspension in scanning electron microscopy is to drop cast it on a flat substrate and then dry it. Here we're looking at an image from a protein nanoparticle solution that was drop casted on a silicon wafer and then dried. What we see is nanoparticles that are in the range of 100 to 200 nanometers, maybe smaller ones, but what we don't have is great contrast, and that's because this solution contains proteins that are in monomer form or in oligomeric form, so it's really a rich solution. One thing that can be done is filtering, and that allows observation of larger nanoparticles in great contrast. And here you can see false coloring on the right. And that morphology and size measurements on these nanoparticles can be made. And if we actually compare the drop casted and filtered nanoparticles, what we see is a similar size, except we can do counting easily with filtered nanoparticles because we have this great contrast. But what we don't have is the native state of this drug product. The way to do that, we have to freeze the nanoparticle solution because we cannot look at it in liquid form in vacuum under an electron beam. And to do that, we have to operate at cryogenic temperatures. The easiest way to do that is plunge freezing, where a drug product is sandwiched between a hat and a carrier, and then quickly immersed in nitrogen slush. And when we do that, we can observe these nanoparticles frozen in almost native state. Except plunge freezing is not a great technique for freezing these types of solutions. And the reason for that is nitrogen does not have a great heat capacity. So what happens is the cooling is not fast enough and ice crystallizes. And when that happens, we see crystalline regions in the microstructure. And these crystalline regions are like honeycomb structures where ice crystallizes and pushes out smaller ions and maybe monomers or smaller particles to the edges. And this almost like a salt brine formation occurs. And this, of course, is not the native state of the drug product, and there is an artifact formed here. So plunge freezing is not really the best way to freeze a sample. Well, what else can we do? What we can do is something called high pressure freezing. When at 2 kilobars, homogeneous nucleation temperature of ice is lowered. And this we can see in the phase diagram of water. And crystallization depends on the cooling rate as well as the pressure. So at synchronized pressurization and cooling, it is possible to obtain amorphous ice where there's no crystallization and the sample is vitrified. That would be a native-like state for this kind of drug product. And that can be done by, again, sandwiching a drug product in between a carrier and a hat. Instead, this time, it's frozen with a high-pressure freezing machine. And there's a freeze fracture step that happens to reveal a surface. There's vacuum evaporation, sublimation, and then afterwards coating of a metal to do imaging under electron microscopy. When we do that with high pressure freezing, we obtain a much different image. What we see is particles that are 100 to 200 nanometers, some somewhat spherical, some highly irregular. It's a very busy solution. It's a very busy image, and that is in fact what this nanomedicine, this nanoparticle colloidal solution is. We compared with plunge freezing, where we see these artifacts, and the formation of these artifacts and larger particles can be attributed to deep etching that is necessary in plunge freezing because of the crystallization, and that can cause formation of agglomerates and as artifacts shown in this image. So when we compare the histograms of these two techniques, we see that a much smaller size distribution in high pressure freezing compared with a much larger size distribution in plunge freezing. That is because we're looking at perhaps a subset of nanoparticles, but there's also some artifacts that are formed during the freezing process. So high pressure freezing is much more complete in terms of revealing the native state of this drug product. Another technique is called freeze substitution. In this case, a sample frozen with high pressure freezing can be brought to room temperature if room temperature measurements must be made, if the cryogenic measurements is not available, or if there are other measurements that can be made on it. And in this case, the water is substituted with another solvent, and then the sample is cross-linked to maintain the structure. When we do that in this false colored image, we can also see these smaller and larger particles, and it's a technique that allows room temperature measurements where the native state of the sample is mostly preserved. So if we compare these cryofixation strategies for the same product, this is what we get. In diameter, in equivalent circular diameter, and in ferrets diameter. When we look at the filtered particle, we see that the particle sizes are slightly larger. That's because we're looking at a subset where the smaller stuff was filtered out. And in all cases, we see that the ferret's diameter is larger than the equivalent circular diameter. That tells us that these particles are not necessarily superspherical. In plunge freezing, that's short for PF here, we see that the particle sizes are large, but also the deviation is large, which can be attributed to the agglomeration that happens during deep etching and the artifact formation due to ice crystallization. In high pressure freezing, we see that the particle sizes are much smaller. Again, the ferrite's diameter is large, but also the deviation in the particle size is much smaller. When we compare that with freeze substitution, we see that the particle size are slightly smaller in freeze substitution. That actually makes sense because this is a room temperature measurement, which means the proteins or the particles here are dehydrated, so we expect them to have smaller size. We can also compare morphology. We can look at the aspect ratios, sphericity, and roundness. And this is the advantage of image-based quantitative analysis in characterization of nanomedicines. We see that high pressure freezing reveals the highest aspect ratio, which means the shapes are mostly irregular, whereas the other techniques do not reveal this. This could be because of filtering of particles. It could be because of agglomeration of particles or other effects. We are also able to compare formulations using these techniques. The subtle differences that may not be obvious otherwise become obvious here using cryo-SEM. We see that different formulations have different sizes. Although these differences are very slight, we see that ferrite's diameter is larger in all cases. In this case, formulation C had slightly less, were slightly smaller. The nanoparticles in formulation C were slightly smaller compared to A and B. We could also compare aspect ratio, sphericity, and roundness of these particles. We saw that formulation B surprisingly had larger aspect ratio compared with the other two formulations. There's also an advantage in multi-attribute characterization. The SEM chambers today have become very powerful. There are other techniques that can be incorporated into this. In this case, we had a Raman probe inside our SEM chamber, and with the Raman probe, we were able to image a nanoparticle from this protein nanoparticle solution and collect Raman spectra from it in its almost native state. And when we collected a Raman spectrum from this nanoparticle, we also compared it with the active ingredient as well as the protein nanocarrier. And when we did that, we saw that the amide band and the CH vibrations, as well as the CC stretching in the active ingredient, were both present in the cryo-Raman. And that gave us confidence that this particle that we saw is actually composed of nanoparticle, protein nanocarrier, as well as the active ingredient. So that brings us to our second challenge question. Which sample preparation method will minimize artifacts in cryo-electron microscopy analysis of an aqueous suspension nanomedicine? A. Chemical fixation. B. High pressure freezing. C. Plunge freezing. D. Filtering and drying. The answer is high pressure freezing. And again, there are two clues here. One is we're looking at cryo-electron microscopy analysis. So chemical fixation, filtering, and drying would not work well. And we're looking at an aqueous suspension where plunge freezing could cause a lot of issues with ice crystallization. So the answer would be high pressure freezing. In summary, nanotechnology has been revolutionizing medicine through nanoparticles that are precisely synthesized to improve the performance of active pharmaceutical ingredients. It's important to understand the limitations of commonly used particle sizing techniques. The take-home message here is that the image-based quantitative analysis offers wealth of information in size and morphology, but to do that, an adequate cryofixation strategy must be used. There's also potential for multi-attribute characterization in native-like state using cryo-SEM. I would like to thank my collaborators, and with that, I will conclude my presentation. Thank you.
Hello everyone. Today I'm going to present on advanced imaging and data analysis to support structure similarity of polymeric formulations. There are two main learning objectives of my today's talk. First, I hope at the end of the talk, the audience will be able to identify what are complex polymeric formulations. And second, through my discussion on GDUFA-funded research, the audience will be able to describe the role of structural analysis to support development of complex polymeric products. Here is an overview of my talk. First, I'll give a few examples of complex polymeric formulations, followed by discussion on imaging technologies and data analysis. What are new imaging technologies for pharmaceutical use and related advanced imaging data analysis. Last, I will provide a short summary to hopefully deliver a clear and helpful list of take-home messages. Here are a few examples of complex polymeric formulations. Based on the properties of the polymer, they can be categorized into non-biodegradable versus biodegradable products. For the non-biodegradable products, there are intrauterine systems, intravaginal rings, and solid implants. And for biodegradable products, there are microspheres, in situ forming implants, as well as solid implants. In terms of generic competitions, right now, FDA has approved one generic intravaginal ring. All others currently do not have any generic competition in the market. Obviously, there is a need to promote development and approval of complex generic polymeric formulations. In the next 3 slides, I will discuss why is generic development of complex polymeric formulations challenging. There are many factors that make generic development of complex polymeric formulations challenging. One of them is formulation composition or the selection of raw material. I'm going to take PLJ microspheres as an example. As shown, our collaborator at the University of Connecticut produced the PLJ formulations using the same method but polymer from 3 different sources with similar inherent viscosity, monomer ratio, and end cap as per the manufacturer's certificate of analysis. The prepared formulations show differences in physical-chemical properties such as particle size, porosity, and average pore diameter, as well as their in vitro drug release characteristics, including burst effect and release rate. Therefore, there is still remaining scientific gap on what are key polymer properties to ensure PLJ polymer with comparable those properties will produce formulations with comparable performance. In addition to formulation factors, manufacturing factors can further complicate generic development of polymeric formulations. Here I'm going to use PLJ microspheres as the example again. As shown, depending on the batch size and or sterilization strategy, the PLJ polymer properties can be significantly altered during manufacturing. Therefore, the PLJ polymer present in the finished formulation may not be very closely represent the properties of the raw material. Accordingly, the remaining scientific gaps are how to figure out what is the raw PLJ polymer used in the reference product, what are the key manufacturing parameters and how do they impact product performance, and when you have successfully developed a small-scale manufacturing procedure, how to scale up without altering formulation characteristics. In terms of bioequivalence approaches, study design for complex polymeric formulations can be a lot more complicated compared to that for conventional dosage forms, because majority, if not all, of these products are designed to offer sustained drug release, and their release profiles can be multiphasic. So here in this slide, I'm using Mirena lipogestural intrauterine system as an example. This product is approved right now for the use of 6 years in vivo. Obviously, a clinical BE study lasting for that long is just practically infeasible. Therefore, this product warrants development of innovative new approach. To date, OGD has posted a product-specific guidance for this one. The PSG recommends 2 studies, in vitro and in vivo ex vivo, based on the product. The generic product has a formulation similarity, comparable physical, chemical, mechanical characteristics, identical dimension. Then its BE can be established using comparable real-time in vitro drug release and a short-term in vivo ex vivo clinical study. So the current approach significantly reduced the burden on the clinical side. However, we do recognize the real-time in vitro release lasting for years can be cumbersome. Therefore, the remaining scientific gap as shown here is to conduct additional studies to fundamentally understand the drug release mechanism and its relationship with formulation parameters and manufacturing parameters, so we could determine whether a properly designed accelerated in vitro drug release can correlate to real-time release that can be used and considered sufficient to ensure bioequivalence. Looking at the audience, I can tell you are thinking, well, generic development of polymeric formulation is complicated, but how can structural analysis help with all those challenges? What is the role of structural analysis? So in the next part of my presentation, hopefully I will address your question. As I have discussed earlier, formulation composition and process parameters can affect the structural characteristics of a formulation, such as morphology, API distribution, porosity, which in turn impacts drug release. Therefore, structural analysis can facilitate reverse engineering to trace manufacturing history and better understand drug release mechanism. Historically, structural analysis of polymeric formulation has been mostly limited to characterization of particle shape and/or surface morphology. This is likely due to the limitations of imaging technologies and related data analysis. However, in the last couple of decades, there has been significant advancement in imaging technologies and related artificial intelligence-based image data analysis. Therefore, nowadays, the structural analysis can cover more and provide in-depth information on API distribution and porosity, which I will further elaborate in later slides. Slide here shows a few imaging techniques that have been used for pharmaceutical applications. Today I'm going to primarily discuss the focused ion beam scanning electron microscopy, FIB-SEM, which is a relatively new imaging tool for pharmaceutical application. Slide here shows a schematic diagram of FIB-SEM imaging station. FIB is a technique used particularly in the semiconductor industry and material science in the past. Nowadays, it has been increasingly used in the biological field as well as pharmaceutical applications. Compared to conventional SEM, FIB has almost identical instrument. It has a dual beam system. The electron and ion beams intersect at a 52 angle at a coincident point near the sample surface, which allows immediate high-resolution SEM imaging of the FIB milled surface. So compared to conventional SEM, the 3D images generated using FIB can provide superior z-axis resolution as high as down to like 3 nanometer. And then because one sample can be in situ milled and imaged, so later when the image data is coupled with modeling and computation tools, the formulation can be reconstructed using the FIB-SEM, therefore to generate in-depth profile through the polymeric matrix to provide information on API distribution, polymer domain, and porosity. Recognizing the potential of FIB and artificial intelligence-based image data analysis, OGD has funded a research project focusing on microstructure characterization with microimaging and image-based analytics to develop a new tool to characterize complex polymer-based long-acting drug products. So in this project, we have selected 2 model systems, including an intrauterine system and a PLJ-based microsphere drug delivery system. In today's talk, I will share some preliminary results to show how structural analysis can be helpful to determine the impact of manufacturing process on physical-chemical properties and in vitro drug release characteristics of PLJ microspheres, as well as how we can develop an image-based model that can be used to predict in vitro release characteristics of this kind of delivery system. The slide here shows an example FIB-SEM cross-section of a PLJ-based microsphere. This image was produced by our collaborator DQMM Solution. As you can see, FIB can generate high-resolution image with information on API particle size, API distribution, PLJ polymer domain, pore size, and pore distribution within a particle. All those information are very helpful to further understand the impact of formulation parameter, manufacturing parameter on formulation characteristics, and correlation between formulation characteristics and drug release. I will further elaborate these points in the next few slides. The slide here shows FIB-SEM sample images of PLJ microspheres. Images A and B are raw FIB-SEM image showing cross-section of a microsphere sample that is 1 year after manufacturing and 2 years after manufacturing respectively. What I would like to highlight in this slide are images C and D, which are artificial intelligence-based image segmentation of the raw FIB-SEM cross-section. As you can see, after the AI analysis, the image segmentation color codes different regions within the particle to clearly show where are API located versus pores versus polymer. So here in images C and D, the green area represents API particles, the red area represents pores, and the blue area represents polymer domain. By looking at those images, you can easily locate the pores and tell if a pore is located in polymer domain only or they are located at the polymer drug interface. Depends on the location and how the pores are connected, their impact on the drug release will not be the same, which I will have another example to further elaborate in later slides. In this slide, I'm going to share some quantitative analysis of FIB-SEM images of PLJ microspheres, which I just discussed in the last slide. The figure here shows 3D API and porosity spatial uniformity of the microsphere samples. The y-axis represents average volume percentage of either the pores or API within the particle, and the x-axis represents the radius of the microsphere. So each data point represents at a location how much percentage volume is occupied by either the pore or the API. As you can see from these profiles, both the API and pores are fairly uniformly distributed across microsphere samples, and the storage time does not seem to significantly change the general distribution of API and pores. In addition to look at the spatial uniformity of API and pores, you could also quantify the particle size of the API and its size distribution, as well as the pore size and size distribution as shown in figure on the right panel. By comparing the porosity or pore size distribution of microsphere sample that is 1 year after manufacturing versus the sample that is 2 years after manufacturing, you can tell the pores slightly decreased during storage. This observation is consistent to the expected polymer enthalpy relaxation, which allow polymer chain rearrangement to likely lead to the observed changes to porosity as described here. In the last 2 slides, I discussed how you may use FIB-SEM and AI-based image analysis to better understand API and pore distribution within a PLJ microsphere and how to quantify their particle size and size distribution. In the next 2 slides, I'm going to briefly discuss how you may use FIB-SEM and AI-based image analysis to correlate those formulation characteristics with drug release. Here I have 2 formulations, F1 and F2. Both were made using the same polymer but different manufacturing conditions. So after FIB-SEM characterization, we have obtained porosity, drug volume percentage, and polymer percentage for both formulations. As you can see, both formulations had similar porosity and drug loading. So by looking at those data, we still cannot tell whether their drug release profiles be similar. But then if you look at their SEM image as shown here, those 2 are raw image data with very high resolution. So if you are an expert looking at SEM images day in and out, you probably can already spot some differences between the 2 formulations in terms of their pore and API distribution. But for people who are not so experienced, despite the resolution is high, it may still be quite difficult. So, but then after the AI-based image analysis transformation, the segmentation images are very intuitive. So by the color codes clearly shows where are the pores versus API particles and polymer located within the particle. So by looking at the AI-based segmentation image, you can clearly see F1 and F2 had quite different pore size as well as pore distribution and API distribution. So by looking at the 2 images, the color-coded 2 images here you probably would guess the two release profiles were likely to be different. Once again, I'm showing you the FIB-SEM raw images and image segmentation of formulation 1 and 2. By looking at the images, even without knowing the exact values for porosity and API particle size, one may guess formulation 2 will likely show higher burst release because more APIs appeared to be surface located or near surface located. Indeed, that was the observation. So the release profiles of formulations 1 and 2 are shown in solid lines here. Formulation 2 had higher burst release, being approximately 30%, whereas formulation 1 only had about 10% burst release. And overall release rate of F2 was also faster compared to the rate of F1. By doing the AI analysis to 3D reconstruct the particles, the research team at DGM also developed an image-based model to predict the release profiles as shown in dashed lines here. So the model predicted release profiles showed fairly good prediction, especially at the initial burst release and the secondary release up to about cumulatively 80% of the drug release. So by doing the image analysis, we were able to summarize what may be primary structural critical quality attributes toward the observed fast release of F2. So the first factor may be the smaller microsphere size of F2, hence increased the surface area. Second, the API distribution heterogeneity of F1 and F2 was different. F2 had more APIs located near surface or on the surface, which contributed to the higher initial burst release. And third, the microporosities and its distribution. So the F2 had more pores across the entire particles, which may contribute to the observed overall faster release. So if you are interested in knowing how to conduct a release simulation, you may go to the references listed here. And if you have questions on details, please feel free to reach out to the corresponding authors listed in this reference for more discussion. In summary, structural analysis can provide insight on impact of manufacturing on formulation characteristics, impact of porosity and API distribution on drug release, and also the analysis can help to better understand what may be mechanism of drug release from the formulation. Therefore, artificial intelligence-based image data analysis is promising to facilitate development of models for predicting drug release, which then can be further explored to enhance in vitro-based bioequivalence approach to facilitate the generic development of complex polymeric formulations. Before closing out my today's talk, I have a challenge question for the audience. My question is, what can structural analysis support generic development of complex polymeric products? A. Reverse engineering. B. Better understand impact of manufacturing on product performance. C. Support development of correlations between formulation characteristics and drug release. D. All of the above. I give everyone 3 seconds to select your answer. 1, 2, 3. Great job, everybody. I see you all selected D, all of the above. Thank you for your attention today. I would be happy to address any questions you may have during the panel discussion.
Thank you to all of our presenters for the great presentations. We'll now enter into our first Q&A panel comprising of our session speakers and introducing Dr. Robert Lionberger, who serves as the Director of the Office of Research and Standards in the Office of Generic Drugs. Dr. Lionberger leads OGD's implementation of the GDUFA science and research commitments, including internal research activities and external research grants and collaborations to ensure the therapeutic equivalence of generic drug products. ORS also provides pre-submission advice on complex generics through the pre-ANDA meetings, product-specific guidances, and correspondence responses. Also, we welcome Dr. Daniel Willett, who's a chemist in the Division of Complex Drug Analysis within the Office of Pharmaceutical Quality. Daniel started his career in 2016 as an ORISE research fellow before becoming a chemist in 2017. While at FDA, he worked on developing a wide variety of spectroscopic and imaging-based approaches in combination with multivariate data analysis techniques for physiochemical analysis of pharmaceutical products on both the nano and macro scales. Now we're moving to our Q&A panel, and we'll be addressing the first round of questions for Dr. Meng Hu. And here will be the first question for Dr. Meng Hu. Can you describe the BEAM tool?
Yeah. Thank you so much for your questions. For this question, so first of all, for those who has no context of a BEAM tool, this word appear on page 7 in my presentation slides. The full name of a BEAM is a Bioequivalence Assessment Mate. Briefly speaking, the BEAM is a data analytical tool developed by OGD based on machine learning, natural language processing, text analysis, those technologies, to streamline all the labor-intensive but less human intelligence involved tasks during the bioequivalence review. So the aim is by just several mouse click, this tool will provide a review report with all the those information field. For example, PK data preparation, table creation, and also routine BE statistical analysis. Those information will be automatically filled by the in the BE review report, so that our reviewer can continue to work on this report, more focus on the human intelligence involved questions. For example, comments and the review. So given the considerable ANDA submission and the tight timeline committed by OGD under the GDUFA, this tool will significantly facilitate the high quality, efficient bioequivalence review to facilitate generic drug development and regulatory assessment. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Hu. And here's the next question. Given so many potentials that the AI models can offer for generic drug development and regulatory assessment, can you elaborate a bit about the limitations of the AI models?
Yeah, again, thanks for the question. I think the AI model has a lot of advantage. We don't need to mention that here. But for the limitation, I see two major limitations for this model. First, it's as we all know, the AI model generally will consider as a black box model, which means we only know the input data and the output data. But for the model structure, sometimes it's not interpretable. This is kind of a two sides of one coin. This setting can make the model can fit all kinds of data structure without any specific assumption. However, in the same time, the model structure will make the model less interpretable. So for some application, for example, face recognition, the AI model, I think it serves very well the purpose, because we basically don't care how they do this. We only care the prediction accuracy. However, if we want to transplant this technology to the drug-related model, we are not only focused on the predictive ability, also we want to know the mechanism somehow. So I consider this is one of the major limit of the model. And also this model, since this is a data-dependent model, they highly rely on the data, the sufficiency and the integrity of the data. It is related to the second limitation I want to mention. It's extrapolation ability of the AI model, because we all know in current stage, the AI model only know what they know. They don't know what they don't know. And although they can solve some complex relationship, but prediction-wise, it's still hard to do the very difficult extrapolation. So one of the potential solution is how can we incorporate the domain knowledge to guide the AI model to conduct more useful predictive tasks. Thanks.
Thank you for responding to that question. We'll now move on to our next panelist, Dr. Miyoung Yoon. And here's the first question for Dr. Yoon. What is the model code required for pre-ANDA meeting package? And does it mean that the detailed program and coding must be used for PK modeling?
Thank you so much for the question. So the clarification is that yes, the model code means the detailed program and coding that to be used for pharmacokinetic modeling. But with saying model code, we also mean that any code you used for generating plots, figures, tables, and also the process you use to conduct virtual BE studies. I hope this clarified. Thank you.
Thank you for responding to that question. The next question for Dr. Yoon. Is MIE approach recommended or practiced only for crossover design, or is it applicable to parallel study design also? And what are the options for parallel study design if MIE is not applicable, which is also popular among LAI products?
Thank you. That is an excellent question. So model integrated approach is aiming to help reducing the burdens of the clinical BE studies for long-acting injectable product. So the challenges with the crossover study remained similar for the parallel design BE studies. So the same strategies with model integrated approach, reducing sample size, shortening study duration, and addressing high PK variability issues with the parallel study design is recommended. So the answer was wrong, but the short answer is yes, the MIE approach is applicable with the parallel design as well. Thank you.
Thank you for responding to that question. We have a couple more questions for Dr. Yoon, and here's the next question. Do you see the MIE approach brought up during protocol development or finalization, or can it be timed before finalizing the statistical analysis plan?
Great question. So I think I commented during the presentation briefly. The model integrated approach and the virtual bioequivalence studies, they can be performed in any time during the product development. In fact, given the complexity of using MIE approach, it can help decisions during early development, and we recommend early communication with the agency. Thank you.
Thank you for responding to that question. We have another question for Dr. Yoon. Would you be able to share your thoughts on leveraging the in vitro data for model integrated BE approaches?
Okay, that is another excellent question. Yes, certainly it's a although it was not the focus of today's discussion, mechanistic modeling such as physiologically based pharmacokinetic modeling, that type of modeling can also be used for model integrated BE approaches. So PBPK model that can integrate product critical formulation attribute such as particle size and their interaction with physiology to predict the drug absorption and pharmacokinetics, so that the virtual bioequivalence studies using those mechanistic modeling and rather predictive in vitro testing, such as dissolution and IVRT studies, can also be used in combination with in vivo PK study result for bioequivalence, or support even in vitro only bioequivalence alternative approaches. Thank you.
Thank you for responding to that question. We have one more question for Dr. Yoon. Where can I find more information about using the model integrated BE, such as examples of MIE for LAIs?
Yeah, thank you for the question. So we acknowledge that this is an area that's rapidly evolving. So we expect that the public workshop I mentioned during the presentation will provide a good opportunity to industry and other stakeholders to learn more about this approach and the associated opportunities. And we are hoping that you all join the discussion on practical considerations when we try to implement the MIE approach, as well as discuss the appropriate ways to validate and verify to provide confidence in using that approach. In terms of the already published resources, we recommend you to check out the publication from our team by Dr. Liang Zhao in Clinical Pharmacology and Therapeutics in 2019, I believe, about generating model integrated evidence for genetic drugs. And another helpful publication to check out is the summary of our team's symposium at the American College of Clinical Pharmacology. The session was about model integrated drug development for long-acting injectable product. So the report has been published in Clinical Pharmacology in Drug Development this year. Thank you.
Thank you for responding to that question. We're going to move on to our next panelist, Dr. Huzeyfe Yilmaz. And here's the question for Dr. Yilmaz. The hydrodynamic radius is different from the dried sample detected in TEM, but the hydrodynamic radius is closer to the real status of the suspension nanodrug. How do we eliminate the difference?
That's actually a very good question. In reality, the dispersed particles are hydrated and solvated, and a hard and a soft corona are formed around it. The composition of the corona, especially the soft corona, is dynamic and fluctuates over time depending on the ionic strength and types of smaller or bigger molecules present in the environment and nature of solvents, the hydrodynamic radius is a measure of the nanoparticle with its corona. In the dry state, this corona may not be present, so there's a size difference between measurements that are performed in dry and frozen states. Rather than eliminating the difference, it's important to realize what is being measured in each case. And actually, this difference is part of the motivation behind the cryo-EM work. We would like to understand how nanoparticles behave in the suspension in their native-like state, which is why we go through all that trouble to fix the aqueous nanosuspension.
Thank you for responding to that question. We have another question for Dr. Yilmaz, and here's the question. What drug product or substance is being represented in your presentation? Can you please identify? Is this an LAI-DP?
So by LAI, I'm guessing that the question is asking if it's a long-acting injectable. So this drug analyzed in the cryo-SEM work was nanoparticle albumin active pharmaceutical ingredient composite, an injectable drug.
Thank you for responding to that question. We have another question for Dr. Yilmaz, and here's the question. What are the main differences between transmission and scanning electron microscopy methods in characterization of nanoparticles?
SEM is scanning electron microscopy is ideal for viewing relatively larger areas, thanks to the larger field of view compared with transmission electron microscopy. With surface images in SEM, bulk structures can be revealed and analyzed. When internal details of nanoparticles are needed at near atomic resolution, then TEM becomes necessary.
Thank you for responding to that question. We're going to move on to our next panelist, Dr. Yan Wang. And here's the question for Dr. Wang. The porosity that you mentioned as an indicator of process variability is not recommended as a critical quality attribute in the product-specific guidance. Do you see that as a requirement coming up in PSG revisions? How do we compare the porosity between ROD and the test product? What's the standard of measurement, as it's quite subjective?
Thank you for that question. It's a very good question. I think, like you said, right now, except the case I mentioned for Marina, all other polymeric long-acting products, the bioequivalence is still established based on clinical studies. So right now, the studies we are doing, the short-term efforts is to explore advanced analytical tools that could be helpful to advance the current methods that are available to facilitate formulation development and improve understanding. So through the improved understanding, then we can start to explore alternative approaches to maybe reduce the burden on the clinical side and put more weight on the formulation characterization. So to your question, whether this would be a requirement or part of the recommendation in PSG revision, right now I do not have an answer to that question because each product specific, each PSG is product specific. So we will have to consider that particular product to see the relevance. In addition, even when we deem porosity is a critical quality attribute for that particular product, as you could see in other similar PSGs, we do not really recommend specific methods. Again, here we're just trying to do some studies to develop new analytical tools for generic companies to choose from. Then the last part of your question about the being subjective, I think the technology itself is not subjective, but I agree in terms of the choice of the particles. I think that's the underlying question here is how do you know the one particle you choose to characterize is representative of the entire formulation? And certainly that is one limitation we do recognize and keep in mind. My today's talk is primarily focusing on FIB-SEM and AI-based image analysis, but when we conduct the research, in addition to FIB, we actually have other orthogonal analytical tools such as X-ray, micro-CT, and conventional polarimetry to confirm and help screening the formulation, then pick the most representative particles or a representative in a range. Thank you.
Thank you for responding to that question. We have another question for Dr. Wang, and here's the question. The porosity characteristics measurement is time-consuming and is done in one microsphere for comparison. How do we ensure that the porosity measurement is representative of the entire lot of the test or RLD? Is there a standard for sampling and measurement of PLGA microspheres for porosity, distribution, and characteristics?
Thank you for that question. And I can see that's one common question from the audience about our expectation for the technology. So like I mentioned, right now we are developing new tools to facilitate formulation development and improve fundamental understanding and trying to connect the dots between formulation characteristics and drug release mechanism. So in terms of porosity characterization, right now, I think that is one actually formulation characteristics most developers would look at some point during formulation development with respect to whether it's the most reasonable to require porosity being comparative. Again, I think we have to put it into a context in terms of which formulation we're discussing. If, for example, we are talking about PLG microspheres, then we know porosity is a critical attribute, but it's not a standalone factor. When we consider similarity or difference in porosity, we have to take other characteristics and drug release into consideration. So here, right now, when we recommend applicants to characterize porosity, both for the ILD and their own test. For the ILD, I think is for you to really have another way to better understand what may be the ILD drug release mechanism, and if you see lot-to-lot difference, what might be the reason. And for characterizing your own test formulation, I think the purpose is really for you to establish a baseline of your own formulation preparation manufacturing. So if down the road, if you ever make a change to your raw material suppliers or your manufacturing procedure, you have a baseline to go back to compare, to save some work. Whether the comparison would be strictly required to establish equivalence, right now we are still at exploratory stage, and we trying to get more data to improve our understanding to make the most reasonable recommendation. Thank you.
Thank you for responding to those questions. The next question will be directed to Daniel Willett, Dr. Daniel Willett. And here's the question. What other emerging technologies are being evaluated for characterization of complex technologies in your lab?
Yeah, so we've currently been focused on evaluating a large variety of imaging-based approaches in our labs within OTR to really take physiochemical characterization to the next level. So this includes the Cryo-EM coupled with Raman that Huzeyfe gave a fantastic presentation on earlier, in addition to the entire gambit of spectroscopic imaging, including laser-directed infrared imaging, terahertz imaging, Raman mapping, and micro-CT. And then these data sets can be further explored by applying multivariate image analysis techniques to the spectroscopic data cubes that are generated, which just allows us to really visualize chemical information within the image. We're also working on taking spectroscopic imaging to the nano region using techniques such as nano-IR which uses the atomic force microscope coupled with an infrared laser to get around no more diffraction limitations that make spectroscopic measurements at the nanoscale challenging. So really just focusing on taking these imaging-based approaches to really physiochemical characterization of our samples.
Thank you for responding to that question. The next question will be directed to Dr. Lionberger. And here's the question for Dr. Lionberger. Can you speak a little bit more about how the outcome of an ANDA submission prediction will be used?
So certainly. So I want to certainly thank everyone for the presentations and thoughtful questions today. So when you think about ANDA submission prediction, there's two levels that it works at. One is on the individual product level, and Meng mentioned a little bit about this, and saying like, how many ANDAs are going to be submitted for a particular product on a particular day, and he focused on the first to file opportunities, which are so important to the generic industry. So that's one aspect of the prediction. The other is at a more aggregated level. So this is something at the level of, if you're thinking of FDA as a sort of business or operational enterprise, how many ANDAs is FDA going to get next year so that we have appropriate staff and appropriate places to review them before they get here. So that's and so you can think of taking the individual predictions and aggregating them up, or at the higher level, maybe there's ways that you can, if you're only interested in the total number, there's ways you can optimize that question. It's a question that's similar to a question that comes up in pharmacokinetic analysis. Do you evaluate the individual pharmacokinetic profiles of a single subject, or are you looking for a conclusion about the population? And so for the prediction algorithms for the ANDA submissions, we're actually interested in both of those cases. And there are certainly internal business cases for FDA on why it's important to have both of those. But certainly this is a technology that, we don't really have any interest in keeping. If we can do this in a better way, we want to share this, because we also think the generic industry in both of those cases could benefit from these and the community as a whole. So talking about the first case, just the individual, when are we going to get applications for a particular reference product? And as Meng said, this goes to some of our internal prioritizations, especially our product-specific guidances. So our goal is to have product-specific guidances available, several years before we think applicants are going to submit their applications, so they can consider the guidances while they're developing their products. And so the more we can predict when the likelihood of these products are, we can attempt to identify the optimal time for the product-specific guidance. It helps us schedule our work to say, well, this one we think applications are going to come in sooner than this one. That may be a factor that will go into our scheduling and our planning, assigning work to particular staff members around that. So that's it. That's an example. But at the workload level, that's sort of at a higher aggregate level. And I think that's something that's very important to FDA, also to the industry, because you want us to have an appropriate number of staff members to review your application in line with the goal dates we've negotiated through our user fee agreement. So, and at that level, certainly that's a very testable prediction. And, I foresee in that space cutthroat competition between different models, right? To say, each year, how many applications are we going to get next year, and which model is the best predictor of that? And I think that sort of, selection pressure will force the models to get better, force people to, evaluate critically what types of tools work better and give you best prediction, what information that you need to collect is most informative to these predictions. And that's why we think it's still a research topic, but there's definitely broad interest in both the predictions at the aggregate level and the individual application level for FDA internally. But I think some of those interests in prediction, if you know how many ANDAs are going to be submitted for a particular NC opportunity, right, that can go into your business planning. Do you think there's going to be 20 applicants, or do you think there's going to be 0, right? Different companies may make different decisions in those cases, but that's the important aspect. And I think what we're trying to do here in our research program is develop the type of algorithms, what types of algorithms and analysis actually work best at this, because we think there's broad value in having good predictions about this.
Well, that's all the time we have for questions in our Q&A panel. We'd like to thank all of our presenters and panelists for answering numerous questions that came in. We'll now enter into our